This is Rochelle, your Microbiology IT tutor. Welcome to today's Microbiology retention session where we will be studying transcription and RNA processing. Now, if you need study resources to help you get that A, then grab your Microbiology IT workbook. The link is in the description box. Transcription and RNA processing. We are going to start with, we have 3 branches, and we're going to start with the first one. And the first one is transcription. And from transcription, we have 1, 2, 3, 4 branches, 5 branches coming out of transcription. We have enzyme, where RNA polymerase is the enzyme. This is sigma factor. So RNA polymerase is the enzyme involved in transcription. We have initiation. Let's move this down a little bit. Initiation, elongation, termination, and then our products. So those are our branches. Going back to initiation, we have the promoter recognition by the sigma factor. So we are going to put sigma factor for short because we don't have a lot of space. Sigma factor. So in initiation for the prokaryotes, we have RNA polymerase hollow enzyme with the sigma factor that binds to 10 to 35 promoter elements. Then we have the eukaryotes. We have the RNA polymerase 2 that requires the general transcription factors to recognize the toxic box. Next, we have elongation. In elongation, we're going to put RNA polymerase synthesizes 5' to 3' RNA. So here on our notes, we have RNA polymerase unwinds DNA and synthesizes RNA in the 5' to 3' direction. The coding strand matches the RNA sequence, except for the U replaces with the T. Next one is termination. And here we have 2 coming from termination. 2. So we have row, independent and dependent. So dependent and then independent. Termination. In prokaryotes. And then in eukaryotes, let's move this over just a tad. In eukaryotes, we have polyadenylation signal. Okay, so with termination, so in prokaryotes, the row dependent uses a helicase to release the RNA. Row independent relies on hairpin structures followed by a poly-U sequence. In eukaryotes, RNA is cleaved downstream of the polyadenylation signal, followed by a poly-A tail addition. The products are messenger RNA, we have rRNA, tRNA, and snRNA. snRNA. Okay, we're going to go to our next branch, and that is domain. And from domain, we have 3 branches: bacteria, archaea, and we have eukarya. So with bacteria, 2 branches from there, we have the single RNA polymerase and polycistronic messenger RNA. In archaea, with archaea, we have 3 branches coming from that. There is 1 RNA polymerase. We have the Tata box and archaeal introns. With eukarya, there are 3 branches: RNA processing. So it's RNA processing. We have introns and exons. And then our last branch is that there are 3 RNA polymerases: 1, 2, and 3. On to our last branch, and it's RNA processing in eukaryotes. And with this one, we have 1, 2, 3 branches. 1, it's 5' capping, methylated guanine, methylated guanine. Our next one is 3' poly-A tail. And then our last branch is the removal of introns. So intron removal. And then exons join. This is splicing, joining via spliceosome. So we have RNA processing, where there is splicing, where the introns are removed, and exons are joined via spliceosome. We have 3' poly-A tail, and then the 5' capping, where the end of the messenger RNA is methylated. It's methylated by the guanine. So we see that here in our notes. The RNA products, messenger RNA, rRNA, tRNA, snRNA, and miRNA. We have RNA processing in eukaryotes only, 5' capping, splicing, and 3' poly-A tail. On to our questions. So number 1, the bacterial transcription initiation factor is called blank. Number 2, a eukaryotic messenger RNA is stabilized at its 3' end by addition of a blank. In prokaryotes, number 3, in prokaryotes, a single messenger RNA coding for multiple proteins is called blank. Multiple choice questions. Number 1, which transcription termination mechanism requires a protein factor? Number 2, which of the following is not true about RNA processing in eukaryotes? So you want to go back to your micro mind map and your notes to help you answer these questions, and then you can put your answers in the chat. Which of the following is not true? So what you're going to do is find the ones that are true and do a process of elimination. Number 3, in archaea, transcription initiation involves, so go back to your micro mind map, find where you see archaea, and then you're able to look at the 3 branches that are attached to assist you with answering the question. And then you can put your answer in the chat. Fill in flow challenge. Fill in the flow of eukaryotic RNA processing. So we have primary transcription leads to blank at 5' end, then leads to blank at 3' end, then blank removes introns, and then mature messenger RNA. Application case. A mutation removes the poly A signal from a eukaryotic gene. Predict what happens to the transcript and its translation efficiency. I'll see you in the next interactive tutoring session. Let's microbiology it.